Thank you, sir.
Okay, welcome back. I start from the first slide itself. And today's lecture is on functions of the cell organelles and the biomembranes. And this sets of slides very comprehensively prepared with 93 slides for 2 hours. Even though there are 93 slides for 2 hours, there won't be enough time to cover all the slides. Maybe we will try our best to do so. And then when you look at these slides, why I made like this, because you are in a lockdown situation, so you may not have access to books and all. So you should be able to stay at home and then again and again go through these slides as well as the recorded version of the lecture. So when you talk about cells, cells are the structural and functional units of living organisms. That is the basic functional unit as well as the basic structural unit of a living organism, right? So the lowest level of structure is capable of performing all the activities of life, we call it. So sometimes we divide these organisms as molecular organisms, microorganisms, unicellular organism, and multicellular organism. When you come to this molecular organism, they consist of molecules only. These molecules are either RNA or DNA and proteins, right? So simple example for these molecular organisms are viruses, right? So in the virus containing RNA and protein is called RNA virus, and then viruses are containing DNA and uh, protein code, they are called DNA viruses. They don't have any ability to do cellular functions, right? And on the other hand, some of the organism we call it as microorganism. These organisms cannot be seen by naked eye, only can be seen through the microscopy. And these organisms are called microorganisms. Examples are bacteria and some of the protozoans comes under this category. Other than that, we have organisms called unicellular organisms. These organisms are uh, uh, examples are there again as bacterias and then amoebas and protozoans comes under unicellular organism, and then the multicellular organism, several cells combine together and then form a multicellular organism that we will come in a while. And then the first reported uh, what you call the knowledge about cell is by Robert Hooke in 1665 from a slice of coke. And subsequently, the cell theory was postulated by Matthias Sladen and Theodor Swan, 1839. What they postulated, all living things are made up of cells. Cells are the smallest working unit of all living things. All cells come from pre-existing cells through cell division. You can understand on the other day when I was doing in the PTP lecture how this eukaryotic cell came from prokaryotic cell, etc. I explained. We'll come to that in a while. And what they say is all cells come from pre-existing cells through cell division. So you may ask me a question: how did the existing cell derive? That is, the answer is given in the PTP. If you go through the PTP lecture slide, you will understand how the pre-existing cell, uh, what do you call that, develop over the period of time. So this was mentioned by Dr. Nisan Sarai yesterday also, the level of structural organization of the human body. So we are starting from atom. Atom forms the molecules, and then molecules form the macromolecules. And then what we say macromolecules in, actually, there are four kind of macromolecules we have basically. These are carbohydrates, lipids, protein, and nucleic acids. And then we consider other molecules are not as micromolecules. These are the important macromolecules essential for the structure and function of a living organism. And then the macromolecules lead to the formation of organelles, and then organelles combine together and then enclosed by a membrane, plasma membrane, and form the cells. And then cells together form the tissues, and then tissues forms the organs, and then organs form the organ system, and then finally the organism. So this is the organizational structure of a human body, starting from atom, and then finally we end up as human body in the organism level. So you can see some of the what do you call that? Basically, cells, right, together form the several cells together form the what do you call that? Tissues. So there are various kind of tissues we come across as we progress through our curriculum, and then you can see some of the cells have been shown here, right? Some of the cells lining of the surface of a tissue, that is epithelium we call it, right? These are epithelial cells, and then epithelium, right? So you can see the characteristic of a epithelial layer, right? In the epithelial layers, you can have the what do you call that? Sometime you can have cilia, and then sometime you can have the microvilli, right? Like in the intestine, you have something called villi for the absorption of food, and then sometimes this epithelial surface you can have the cilia, right? In our respiratory tract, you can have cilia, and then uh, and then you can see the when you look at this, they have the narrow extracellular space. They are stacked together, compactly stacked together, and then they have the nucleus and other cellular organelles. And then other than that, they have what do you call that? Cytoskeletons they have, and then they have having supplied by the nerves, right? And then capillary, that is what you call that beneath the epithelium, you have what you call that another tissue called connective tissue, right? And then you can see what you call that there is a between the epithelial layer and the what you call that the connective tissue, there is a basal region we call it. There, that is, you have the basal lamina, right? And then basal lamina and the reticular fibers called form together, they form the basement membrane. And then you can see in between the cells there are several cellular junctions. We will be talking about this later on. And these cellular junctions are tight, can be a tight junction, it can be an adhesive belt, or it can be a desmosome, it can be a gap junction, right? And then what happened, the transport transcellular what do you call the transport is controlled by these junctions, cellular junctions. Okay, we will be dealing about this in detail later on. And then when you come to this type of tissues, another type of tissue is called connective tissue. And then there are a lot of connective tissue, even include the blood itself, it's a connective tissue. And then uh, these connective tissues can be collagen based or elastin based connective tissue. And then we will be talking about more in the extracellular matrix. And then you can see there is another tissue called fat cells or adipose tissue. Mainly these cells consist of what called fat, right? Beneath the skins, what happened, this adipose tissue you can find, and then they are the ones storing the fat in the body. And then cartilaginous tissue, we call it as some of the areas in our body is called cartilage. And then these cartilages are located in certain areas, and then this is the cartilaginous tissue. And then again, you can see the calcification of the cartilaginous tissue will lead to a bone tissue. So the difference between the cartilaginous tissue and the bone tissue is shown in the picture. As we go, you can see that the type of tissues, what is called that muscle tissues. And then I'm not talking much about this. We will be studying in the body tissues and locomotor system. There are two kind of muscle tissue you have. One is called skeletal muscle, other one is called smooth muscle, and other one is called cardiac muscle, right? The muscle located in the heart or the myocardium of the heart is called cardiac muscles. And then some some muscles are called smooth muscles, and then some muscles are called that the muscles connected to the bones, which are usually skeletal muscle we call it. And then you can see that another tissue is shown in your right hand side, that is what they call, when you look at through the microscope, that is the appearance of the nerve tissues. And then you can see the neurons, and then the continuation of the neurons are the accents, uh, accents of what do you call that, neural tissue. Okay, anyway, now you can see from these tissues, we can see what happened, the development of organs taking place. Now, simple example you can see now, if you look at our upper arm, right? So when you look at the upper arm, right, there are various tissues are located in this, right? You can see there is a fibrous connective tissue that is forming the tendon, right? So you can see the tendons are connecting the bones and the muscles, right? And then you can see there is what you call that loose connective tissue under the skin. There is another connective tissue under the skin. And then you can see the fat droplets again beneath the skin, adipose tissue. And then you can see again the cartilage tissue that is in the joints. You can have the cartilage tissue, right? And then you can see the blood supply to the upper arm, upper limb. And then there are a lot of cells in that blood. They're considered as again connective tissue. Red blood cells will be there, white blood cells will be there, plate cells will be there. The liquid portion of the blood is called plasma. Understand? And then you can see there are 3 bones shown there. That is what you call bone tissue. So all these combined together, they form what you call that organs, isn't it? So we can see that there is a fibrous connective tissue, there's a loose connective tissue, there is a blood, it is itself connective tissue, adipose tissue is connective tissue, and cartilage is connective tissue, and then bone is again another tissue, isn't it? So this is combined together, several tissues combined together form the organs. On the other hand, when you look at the right hand side, you can see the stomach, right? And then that is again considered as an organ. It composed of several tissues. And then you can see the epithelial tissues, right? And then lining, the epithelial tissue is the one lining the stomach wall, and then it involved in transport and secretion and absorption, whatever comes into the stomach. And then subsequently, you can see the beneath that there is a epithelial tissue, and then you can see the connective tissue that is giving the support, strength, and elasticity. And then you can see the beneath that another layer called muscle tissue that is for the movement, right? The churning activity of the stomach, right? When the food is being taken up by the stomach for digestion, right? There's muscle tissues involved, and then the nervous tissue also there. That is information processing, communication, and control is taken by. So this is what you call that an organ formed by several tissues. Example for organ formed by several tissues. Okay, so we are starting from the atom. Atom become molecules, and molecules become organelles. Organelles become cell. Cell become tissue, and the tissue become organs. And then subsequently, you can see these organs combine together and form the organ system, right? So we have several systems in our body. One is, we call it as lymphatic system. It consists of lymph nodes, right? And lymphatic vessels, right? And then there is, this is the one usually involved in the defense against the pathogens. Nowadays, we are talking about the COVID, and then immunity, and then immune boosting, and then vaccination, and then all these actually, anything enters our body is called antigen. Our body produces antibodies. That is what you call antigen antibody reaction take place in the body. And what happens? The lymphatic system, right? It is mainly together with the immune system defense against the pathogen. So you can see that there is a gland is there that is called thymus, and the lymph nodes, right? And spleen, and this is what you call lymphatic system. And then you can see, you will be studying later on, one is called another organ system called respiratory system. That is nowadays is very critical, isn't it? Said most of the people trying to protect their respiratory system from COVID, but COVID doesn't what do you call that, respect any of you, and then that will infect and sometimes kill you, isn't it? So you have the removing the carbon dioxide and inhaling the oxygen. So nowadays the oxygen saturation, oxygen cylinder, and then so that is the talk of the town. So that is what you call respiratory system, right? Nasal passage, trachea and lungs, and the details will be taught in the system itself. And then we come to the digestive system. That is what you call that process of the food for the use by the body, and then remove the waste. The undigested food as removed as a waste, and then it consists of oral cavity, esophagus, and then you get the stomach, and then subsequently the intestine, small intestine, last intestine, colon, and rectum. And then this is what you call, and then associated glands such as associated organs such as liver, gallbladder, etc., etc. So this is what you call digestive system. So the organs combine together, organ system or digestive system now. And then the urinary system. The urinary system again control the electrolytes and water balance in the body, and then not only removes the what do you call that, water balance is whether we are in a dehydrated condition or whether we are in a hydrated condition, and then how these our electrolytes are balanced, right? That means we are our osmol the potassium, sodium ions, there's excretions and absorptions, all these controlled by this urinary system. And then control the water balance in the body, remove the waste, right? When you say the waste is then something like the urea is a waste product, isn't it? And then the urea very high level in the blood is very toxic to the brain. And then sometimes we get creatinine. All these are nitrogenous uric acid. That is again another compound. See, this is a nitrogenous compounds excreted in the urinary system. So that is also evolved from the organs become what do you call that system, right? And then come to the reproductive system. So the male reproductive system and the female reproductive system. Actually, the reproductive system is given for the what do you call the generation. What do you call that? What do you call the reproduce, and then uh, uh, okay, the male reproductive system that is produces sex hormones, gametes, and then delivers gamete to the female, and then in the female reproductive system, the sperms are right uh, uh, produces sex hormones and gametes, and then there is fertilization taking place inside the female reproductive system in the fallopian tube that you would have done in your A level. And then what happens, you get the formation of the ovum, and then that is embryo, and then that is developed into a fetus, and subsequently you get the child. So this is what you call the reproductive system. Okay, so that is what I recap again. What atoms become molecules, and molecules become what do you call that, organelles become cells, and cells combine together form the tissues, and then they are the differentiation of the cell, we call it actually, right? So cell, when they differentiate, and then they combine together, and then they form the tissues, and then tissues become organs, and then organs develop into organ system, and then organ system combine together form the human body. So this is the structural organization of the human body, which you will be studying for the next 5 years in a very, very different levels. You will be talking about it. Okay. So when you come to the structural, we are talking about the cell now, right? So for all these introduction is to start with the cell, right? So when you look at the cell, right, you can see there are this organization of the cell. All these time I talked about the organization of the human body. Now I am talking about the organization of the cell, right? So you can see there are 4 levels we come across. One is we call level 1, level 2, level 3, and level 4. Right? The level 1 is the monomeric units. That's what I said. These monomers combine together. They what do you call that? They polymerization, or they combine together by condensation reactions and form the micromolecules. Right? So most of the time, right, from the monomer, we get the polymer, right? For example, yesterday, Dr. Nisansala taught you something about, you can see from the glucose is a monosaccharide, it can become a polysaccharide, right? Starch and glycogens are polysaccharides. When you have the amino acids are the monomers, and then they have combined together and then form the micromolecule protein. and then you can see that is what you call fatty acid and then glycerol combined together form the triacyl glycerol. So that is from the level 1, we go to the level 2, the micromolecules. The 4 major micromolecules are proteins, carbohydrates, lipids, and nucleic acids. And then from there, you can see the supramolecular complexes. That is what I want to talk about. Yesterday, one of the students asked is what is supramolecular complex? Actually, you can see now, right? The DNA, right? When the DNA is actually all the information is stored in the DNA, as Dr. Nisansala yesterday said, why we have a DNA, why we have a RNA, right? So there is a reason for that, okay? So we have the central dogma. In the central dogma of molecular biology, when you take DNA produces RNA, and then RNA produces protein. That is what you call DNA become DNA replication, DNA become RNA is called transcription, RNA become protein is called translation. And then, so the DNA is a larger molecule, then the information in the DNA has to be given to the cytoplasmic compartment for the protein synthesis. The DNA cannot come out. So that the message is printed in a smaller molecule. That molecule is mRNA. That's we call messenger RNA. But when you look at the DNA, right, and then it is located inside the nucleus as the chromosome. The chromosome, we call it as a supramolecular complex. Why we call supramolecular complex? To accommodate in a similar structure like the nucleus, right? What happened? There is a process called condensation taking place, right? The chromosome combined together with what you call that histone, right? So when the proteins associated with these are called nucleoprotein, we call it, and then you can see the nucleic acid combined together with the protein, and then it forms a supramolecular complex. So example for the supramolecular complex is what you call that chromosome. Understand? Right. And then you can see the various levels. That is about the nucleotide. Then you can see amino acids, and then amino acids become protein, and then proteins are the functional component of the plasma membrane embedded in between the lipid bilayer. And then you can see another one is sometime in a plant cell, there is additional membrane is there, that is we call cell wall, and then that is made by another polymer of carbohydrate that is called cellulose. Okay. So all these, what you call, that is a molecular organization of a cell, right? How this molecular organization of cell took place. You can see every organelle inside the cells, these molecules are involved. And then not only that, the membrane covering the all these organelles and the form the cell also made out of what? Protein, lipids, carbohydrates. So what this is what we call the different structural organization hierarchy, structural hierarchy in the molecular organization of a cell. Level 1, monomeric units. Level 2 is micromolecules. Level 3 is supramolecular complexes. And level 4 is the cell and its organelles. Okay. And then when you go to some organisms, single cell, as I said, mentioned earlier, they are called unicellular organism. Other cells are multicellular aggregates of specialized cells. Whether multicellular or unicellular, all organisms must accomplish some same functions. That is what you call that uptake and processing of nutrients, and then excretion of the waste products, and response to environmental stimuli and reproduction, among other. That is simple, right? So you have to live, right? And then you have to eat, you have to excrete, and you have to reproduce. That's also we are doing, isn't it? All these MBBS, PhD, and all these are reproduce our generation, isn't it? That's the idea of it, isn't it? So we study, and then we do a job, and we get married, and then we get married, we have children grow up, and then they study, they do job, and then they get married, they have their children. So it's a reproduction, isn't it? Every organism wanted to reproduce its generation, isn't it? That is happening. So uptake and processing of nutrients, you think in your words, right? You compare you to the cell. Cell also has to do that, uptake and processing of nutrients, excretion of waste, yes, of course, and then respond to environmental stimuli and reproducing, among other, right? So that is a basic functions of a cell. Okay. And then cells are not, all the cells are not same, isn't it? So you can see we are having 130 people are here in this days. You can see some people are short, some people are tall, some people are, right, what do you call that, fair, some people are dark, and then okay, little different, right? So we are not same, right? Cells are not same. So cells with they also have different shape and size, right? So you can see some of the cells how they appear. I just wanted to show you, right? Not actually this is not to by heart or anything like that. You can see the first one. You can see the eubacteria, the appearance of the eubacteria, and then you can see the second one is the archaebacteria, and then you can see some of the blood cells, right? White blood cells and red blood cells, how they appear, platelets. And then you can see the dinosaur egg is a large single cell, right? And then you can see the single cell green algae. And then you can see the neuron of the cerebellum. This is what you call Purkinje's neuron of the cerebellum, how it looked like. That is again a single cell. And then you can see epithelial sheet, how these epithelial cells are stacked together in a epithelial tissue. And then you can see again some of the plant cells also. I have shown you that to differentiate that cells from the animal cells. Okay, so cells with different shape and size. Okay, all the organisms in nature or in the globe, right? They are under three distinct domains. These domains we call it as kingdoms or domain, right? So one of the thing we call it as archaebacteria. Archaebacteria, there are extreme halophiles. Halophiles mean that salt liking, right? Bacteria. Methanogens. Methanogens means the bacteria produce methane, right? And thermophiles, and mean extreme heat condition. The bacteria who can survive in the extreme heat conditions, the thermophiles. So these comes under archaebacteria. From these archaebacteria only, the evolution took place, and then you get the eubacteria. The eubacteria is Thermatoga, flavor bacteria, cyanobacteria, purple bacteria, gram-positive bacteria, green non-sulfur bacteria. All these together we call it as prokaryotes, right? And then you can see there are what do you call another domain we call eukaryotes. Most of the time these eukaryotes are multicellular, right? Animals, right? And then fungi, plants, and then you can see the organism which are having cilia. The organism which are having cilia, you would have done in your A level, paramecium, right? And then flagellates. There are a lot of bacteria having flagella, Euglena having flagella, and then another type of bacteria is called microsporidia. So that is you don't need to individually know this. Idea is what? There are three domains we have: archaebacteria, eubacteria, eukaryotes. Archaebacteria and eubacteria considered as prokaryotes, and then others are considered as eukaryote. That knowledge is adequate enough for you. So when you come to this, the two large groups of prokaryotes can be distinguished on biochemical grounds. That's what I am teaching here. Archaebacteria and eubacteria, right? Eubacteria inhabit in soil and then surface waters and tissues of other living or decaying organism. You can see that. There's a MCQ point is there, right? Most of the well-studied bacteria, including Escherichia coli, are eubacteria, right? On the other hand, archaebacteria, more recently discovered, are less well characterized biochemically. Most inhabit extreme environments, salt lakes, hot springs. That's what I say, halophiles. The halophiles mean, halophilic means salt liking. Hot springs, they okay, uh, high temperature and high acidic box, and then ocean depth. This is called archaebacteria. That means the bacteria who can survive in the extreme conditions, we call archaebacteria. And then bacteria lives in the normal surfaces are called eubacteria. And then so you can see now, we not only we divide these as kingdoms and domains, and then we can divide these, what do you call that, organism according to the source of energy, right? So you can see some of these organisms are called phototrophs, energy they derive from the light. Hemotrophs, the energy derived from chemical compounds. So when you look at the phototrophs, right, they assimilate carbon dioxide into nutrient, right? Examples are cyanobacteria and plants, right? And when you come to heterotrophs, they are deriving the carbon from the organic compounds, purple bacteria, green bacteria, right? So those are heterotrophs. Heterotrophs is energy from chemical compounds. In the heterotrophs also, we have heterotrophs, carbon from organic compounds, and then we divide them into two, lithotrophs and organotrophs, right? The energy from inorganic compounds, sulfur bacteria, hydrogen bacteria, they are called lithotrophs. And from inorganic compounds, they derive energy. Organotrophs mean they are usually getting from organic compounds. Most of the prokaryotes, all the non-phototrophic eukaryotes, right, comes under organotrophs. So this is how we divide it. You can see organism based on the source of energy. Understand? Okay, right. All of you okay now so far?
Okay, sir.
Are you all there or sleeping?
No, sir. Okay, sir.
Okay, right. So now we can see basically cells can be divided into two categories. One is we call prokaryotic cell and eukaryotic cell, right? So the electron microscope appearance of a prokaryotic cell is shown you on the top, right? And then the eukaryotic cell also shown you in your right hand side. So the basic structure of a prokaryotic cell is different from the eukaryotic cell. The fundamental difference is what you call that, the DNA or else the genetic material of the prokaryotic cell is usually located in the cytoplasm. It doesn't have a membrane structure. Then that's called, we call nucleoid, isn't it? And most of the organelles lacking in the prokaryotic cell, right? And then it has a inner plasma membrane, and then it may have a cell wall, and then it may have a periplasmic space, and then it may have a outer membrane, right? And on the other hand, when you look at a eukaryotic cell, the fundamental structure of a eukaryotic cells, the genetic material is covered by, right, what do you call that, membrane, that is we call nuclear envelope or nuclear membrane, and then the DNA is associated with the histones, and you have organelles. Plasma membrane is the membrane enclosing the, what do you call that, organelles and the cytoplasm. There are several organelles. We will be going into detail in a while. Golgi vesicles, mitochondria, peroxisome, lysosome, right, secretory vesicles, rough endoplasmic reticulum, smooth endoplasmic reticulum, all blah blah will be there in this eukaryotic cell. So this is a fundamental difference between the eukaryotic and the prokaryotic cell, right? So when you come to this, we take the prokaryotic cell in detail bit, okay, right? So what we take the prokaryotic cell, we take one of the example for a prokaryotic cell is the bacterial cells, right? So you look at a bacterial cell, standard bacterial cell in your left hand side, right? There you can see there are, right, what do you call that, this surrounding the bacterial cell, there are, what do you call that, what do you call, projections, we call a flagella, right? That is propelling the cell through its surrounding. And then you have the cell envelope, and then the provide points of addition to the surface of the other cell, that's called pill. And then you can see the genetic material is called nucleoid, contain a single, simple, long circular DNA molecule, right? You can see that. And the ribosome is there to synthesize the protein. And then other than that, you don't have mitochondria, you don't have endoplasmic reticulum. Understand? All these organelles are not there, right? It's a very basic structure. Ribosome is there for the synthesis of protein. And then when you look at this, you can see, sometimes you might have studied in your A level, some bacteria are called gram-negative bacteria. Some bacteria are called gram-positive bacteria. You don't know why we call gram-negative bacteria, why we call gram-positive bacteria. So there is a gram stain. So when you use the gram stain, it is stained by the gram stain, then it's called gram-positive bacteria. It's not stained by the gram stain, it's not called gram-negative bacteria. Simple as that. But there's a fundamental difference, right? You can see now when you come to the gram-negative bacteria, there are three layers you have. One outer membrane and the peptidoglycan. The bacterial cell wall is known as peptidoglycan layer. And then you have another membrane, inner membrane you have. So that is what you call gram-negative bacteria. Outer membrane is peptidoglycan layer, and then outer membranes are there. And when you come to gram-positive bacteria, the outer membrane is not there, right? Only the peptidoglycan layer and the no outer membrane. The peptidoglycan layer is thicker than what do you call that? Gram-negative bacteria and gram-positive bacteria, right? And then you come to this cyanobacteria, is gram-negative. It has a tougher peptidoglycan layer, right? Internal membrane system with photosynthetic pigments. Okay, you can see that cyanobacteria comes under phototrophics. So phototrophics mean they have the ability to synthesize own nutrients, right? Because of this, what do you call that? Photosynthetic pigments are there in cyanobacteria. And then when you come to archaebacteria, that is what you call that they live in extreme conditions. I told you, no outer membrane, peptidoglycan layer outside, and they have a plasma membrane. So this is the common structural features of a bacterial cell, and then why we call a bacteria gram-negative, and why we call a bacteria gram-positive, and what is cyanobacteria, what is archaebacteria, and what are the differences in their, what do you call that, membrane, cell membrane or outer membrane. Okay, right. So this is what we are now coming to the eukaryotic cell. There are eukaryotic cell. There are two kinds of eukaryotic cell. We can have the cells are in the animals and the cells are in the plants, isn't it? So when you look at a standard structure of an animal cell is there is a nuclear envelope, and then inside the nuclear envelope, you have a DNA and protein, and then you can have the plasma membrane, separate the cell from the environment, regulate the movement of substances across the membrane, and then you can have what you call the rough endoplasmic reticulum originating from the nucleus and spreading all over. And then when you come to this inside the cell, there is another one is called nucleolus, and which is having RNA. That RNA is made out of protein and nucleic acid, that is called ribosomal RNA. And then when you come to the outside the nucleus, there are smooth endoplasmic reticulum, which is involving in the lipid biosynthesis and drug metabolism. And there is a, what do you call it, stacked vesicle-like structure we call Golgi complex, right? And then that is involving in the protein packages and then targeting the protein to other part of the cells or other part of the body. And then you have what you call that transport vesicles, shuttling the materials from whatever synthesized in the ribosome, sorry, rough endoplasmic reticulum or smooth endoplasmic reticulum, shuttle through this vesicle to the, what you call that, Golgi apparatus. And then you can have the suicide bag, right? Suicide bag is lysosomal degrades intracellular debris. That is what you call that. We call it. Sometimes you can see that that for example, we have a what you call mitochondria. We have mitochondria is that when it's matured mitochondria, then it's no more useful. Then what happens? it is destroyed by the lysosome by a mechanism called autophagy, right? We will be studying later on. And then some of the proteins, right? That means the proteins, some are, what do you call that, modified proteins, and then that is dangerous to the cell. They are removed like that. And then sometime what happen, we have, what do you call that, proteins are no more use, then they are also removed by this lysosome. And then the cell death, later on necrosis of the cell, also through the leakage of, what do you call that, lysosomal enzyme, right? So that is what you call suicide back of the cell, we call it. And then you can see cytoskeleton support the cells, right? And then each and every cell have a structure. The structure is actually basically because of the cytoskeleton. And then imagine, you can see, uh, imagine we don't have a cytoskeleton, right? So suppose we don't have a skeletal system. When we don't have a skeletal system, what would have happened? We are like, what do you call that, mass of, what do you call that, some muscle adipose tissue, something like that, right? So when you are looking at a medical lecture, you can see various muscle masses will be in the on the, what do you call that, chair, and then the lecturer also will be like a muscle mass, isn't it? Then we have to lift and see, oh, who is this? Is it Dr. Jiva? Is it Metma? Is it Lima? Is it Imesha? Like that. So our structure, right, is given by our body structure, right? Is given by what? Our skeletal system, isn't it? Okay. So the skeletal system not only give a structure, but also involving in the movement of the organism, isn't it? Same thing when you look at the cell also, the skeletal system located inside the cell is called cytoskeleton. And then we will be, in the next lecture, I will be teaching you about this. And then you can see, so a lot of machinery, it is that which the work is distributed, isn't it? Work is distributed among different organelles. And then you can see the mitochondria, that is the energy house of the cell, and ribosomes involving protein synthesis. So there's a slight difference between the animal cell and the plant cell. And then you can see the plant cells, additionally, you have, what do you call that, chloroplast will be there. The chloroplast is the one involving in the synthesis of, what do you call that, carbohydrates. And then you can see the starch granules as, and then stored there. And there are some membranes called thylakoids, right? They are the sites of light-driven ATP synthesis. And then you can see additional to the plasma membrane, it has a cell wall. And then there's a slight difference. You can see the vacuole is there, right? The vacuole is not there in the animal cells. And then, right, cell wall, as well as Golgi complex, ribosome, ER, everything is same. Even cytoskeleton also present in the plant cells. So there's a different. I just wanted to show you the difference between the animal cell and the plant cell. So a cell has to do a lot of work, isn't it? A cell has to do a lot of work in a sense. What are these works? Right? So when you get up in the morning, you have some work, isn't it? You have a Zoom lecture, you have to eat, and then you have to go for marketing, and then you have to text your friend, or so you have a lot of work, right? So cell also has a lot of work, right? So cells build and degrade numerous molecules and structure. So that is what you call that, right? These metabolic processes only can happen in the cell, right? The molecular organism doesn't have these metabolic processes. When you say metabolic processes, what is metabolism is, right? All chemical reactions taking place in the cell is called metabolism, right? So we categorize the metabolism into two: anabolism, catabolism. The synthetic reactions are called anabolism, and the degradation reactions are called catabolism. So what happens? Cells continue to degrade and synthesize numerous molecules. Understand? Okay. And produce their own external environments and glue. Right? Each and every cell surrounded by an external environment, that is what we call extracellular matrix, we call it, right? And then we will be talking about in our body tissues and locomotor system. This extracellular matrix is actually produced by the cell. That means the cell can produce its own external environment, we call it. I will be dealing with in detail later on. And then cell can change shape and move. Understand? Right? The simple example you can see, macrophages can change their cell and then they can move from one place to another place. Right? For example, if we have an infection or inflammation in that area, that macrophages will be recruited, monocytes will be recruited, and then there what happens? These cells are moving. Right? And then cells can sense and send information. That means they can sense a stimulus from the environment or surrounding or from another part of the body and then can send information. And then cells regulate their own gene expression to meet changing needs. What protein has to be synthesized, in what amounts has to be synthesized, where these protein has to be sent. Understand? So this is all gene expression. Understand? Simple example I am telling you, insulin is a protein. When we eat a heavy carbohydrate diet, when I say heavy carbohydrate diet means half a loaf of bread and one potato curry, one dal curry, finish. That is a heavy carbohydrate diet, isn't it? So immediately what will happen? Our blood glucose will go up. Oh, so what happened? That is detrimental. So we have to bring it down, isn't it? So what happened? There is a cell called pancreatic beta cell that senses the glucose. Whatever the glucose level goes up, it senses. So what it does, right? So it synthesizes insulin and sends into the blood to other target tissues. Then what happens? Other target tissues will take up the glucose and metabolize, and glucose level will be brought down. So cells sense and send information, right? And cells regulate the gene expression. So at the time when the blood glucose increases, insulin gene, right, produces what? mRNA. mRNA produces what? Insulin protein, isn't it? Gene expression. There is, and then you can see, other than the beta cell of the pancreas, insulin cannot be produced in any other cell. So this is a simple example for gene expression. So how did we synthesize a artificial insulin, recombinant insulin? What we did from the human beta cell, we extracted the DNA for the insulin gene responsible for insulin, and then we took it and then we put it in the E. coli bacteria. In the E. coli bacteria, there's a DNA called plasmid, and then we fuse this DNA, insulin DNA, with this E. coli DNA. What happened? That when the E. coli bacteria multiply, that insulin gene also multiply, that is ligated to the plasmid, and then that will express in the E. coli and then produce insulin. That is what you call recombinant insulin. So that is what you call gene expression. Understand? So we can make gene expression happens in some other tissues. Most of the time, that is very helpful in the drug development, this gene expression. And then you can see cells grow and divide. This is what we do. This will be done in anatomy as well as in biochemistry. One of the lecture called cell cycle, right? So you can see now a cell grow and divide, and after that the cell die, isn't it? Should die, right? So what happened? Cell can suddenly die. When a cell suddenly dies, that process is called sudden death. It's called necrosis. Understand? Or else the cell has to be, right, programmed to die. That is called programmed cell death. The programmed cell death is called apoptosis, right? Okay. Sudden cell death is called necrosis. So cells grow, divide, and die. So that is controlled, regulated by a mechanism called cell cycle. Understand? If the cell cycle is not regulated, cell continue to divide, cell continue to grow. That is end up as cancer, isn't it? Okay, so that is all happening. And cells die from aggravated assault or an internal program. That's what I said. Aggravated assault means that, for example, that sometimes a simple example, heart attack, right? You all studied your A-level knowledge of what you call that heart, isn't it? We call it as the ventricle muscles is called myocardium, isn't it? That is supplied by, there are two vessels coming down that is called coronary arteries, and they supply the blood to the myocardium. And then what happened? We are having a normal aerobic tissue. Heart is a aerobic tissue. Understand? Our habits, our bad habits, we eat a lot of fatty foods, we smoke, we are under stress. What happened? These arteries which are supplying the blood to the myocardium can be narrowed down. When it narrowed down, what happened? The blood supply to the heart myocardium decreases. When the blood supply to the myocardium decreases, mean oxygen supply decreases. When the oxygen supply decreases, mean ATP decreases and the cells die. So that is what we call aggravated assault, we call it. And then that condition is called necrosis. So when the cells die in the myocardium, you feel severe chest pain, and then part of your myocardium is gone. That is what you call myocardial infarction. They use the medical term, and then layman point of view, we call it heart attack. And then you can see there is an internal program, as I said, when the cell is divided, it is controlled by a lot of signal transduction mechanism. And then there, that's called cell cycle regulation, we call it. And then what happened? A cell is programmed to die after some time. That is what we call programmed cell death. We call it as apoptosis. Apoptosis, A-P-O-P-T-I, apoptosis, T-O-S-I-S. Apoptosis means programmed cell death. Sudden cell death is called necrosis. Okay. So as I mentioned, what happened? Cells build and degrade numerous molecules and structure, as I said, isn't it? So some of the reactions are synthetic reactions. You need ATP. And some of the reactions are, we call it as catabolic reactions. So the ATP derived from the catabolic reactions are utilized in the synthesis of macromolecules from smaller molecules, right? That is what we call the difference between the catabolism and anabolism. So cells degrade and then synthesize, right? For example, amino acids are available, protein is synthesized. When the protein has to be degraded, it degrades into amino acids, right? When the glucose is available, glycogen is synthesized and it is stored in the liver, isn't it? Why we have to store glycogen in the liver? Yes, when we don't have a food, that glycogen can supply the glucose. Understand? Suppose you have glycogen in your liver, right? You had a fight with your mother, right? And then you wanted to skip your dinner. So your mother is asking, right? Dua, come and eat, puta, come and eat. And you said, I don't want. Why you don't want? You know, your liver has glycogen, isn't it? So that will supply your glucose overnight. Your mother doesn't know biochemistry, you know, isn't it? The same thing. Like, so it stores the glycogen, and whenever needed, the glycogen is utilized. Understand? So that is what we call anabolism and catabolism. Catabolic reactions will, whatever the ATP produced will be utilized in the anabolic reaction. So what is called synthetic reactions, other one is we call catabolic reaction, breakdown reactions. Okay, why? Right. So why is ATP needed? Synthesis of micromolecules like DNA, RNA, protein, polysaccharide. And then why we need ATP? Synthesis of cellular constituents like that, membrane, phospholipids, and certain metabolites, and cellular movements, you need ATP, right? So muscle contraction, crawling movement of cells, right? Movement of chromosomes during mitosis and meiosis. All these are movement, isn't it? And then the proteins or substances synthesized in the cell, in the endoplasmic reticulum is transported to the Golgi apparatus, right? It's a movement. And then what happened? From one cell, one hormone is synthesized. For example, pancreatic beta cell, insulin is secreted, and insulin is secreted into the, what do you call that, blood and then transported to the muscle. Again, what transport is involved, isn't it? So transport of molecules against concentration gradient, sometime you need energy, right? And then electric potential across the membrane, the nerve function, right? So once you keep your finger in the what you call fire, you suddenly pull it out. So why there is a stimulus and the response. You need what? There's a nerve function is needed, isn't it? That is what do you call that? Nerve conduction. Okay, so you can see. So some process you need ATP, and then some process produce ATP. That ATP utilized by the process. Okay, so now what happened there? The surrounding environment, very careful. The surrounding environment of a cell, we usually call it as extracellular matrix. We call it in shortened form, we call it as ECM. It is a 2-hour lecture I will be teaching you. And then you can see there's an epithelial surface. You know what is epithelial tissue now? You know they are stacked together very closely. And then you can see beneath these epithelial cells, there is a membrane-like structure, right? That is what you call basal lamina. And below the basal lamina, you have what you call that, our connective tissue. This connective tissue is the one we call it as extracellular matrix. When this connective tissue has collagen fibers, macrophages, blood supply capillary, there are cells called mast cells, and sometimes you have the elastin fibers, elastic fibers, and there are some of the molecules called hyaluronic acid, proteoglycans, glycoproteins, etc. I will be doing this in the extracellular matrix lecture in detail. What is hyaluronic acid? What is proteoglycans? What is glycoprotein, etc. But you have some idea. Cells are attached to the environment. They glue to the environment via a basal membrane or basal lamina, right? And then beneath the basal lamina, you have extracellular matrix. Okay, so epithelial surface, then there's a basement membrane or basal lamina, and then beneath that you have the connective tissue or extracellular matrix. Okay, there's another function of the cells. Cells can change shape and move. This is interesting, isn't it? So you can see the movement of a cell. Within the cell, there is movement take place. Cell itself can move, right? So you have what do you call that? There are filaments. That is the next lecture. That is called cytoskeletal filaments. We have 3 kinds of filaments we have. Intermediate filaments, microtubules, and microfilaments. Understand? So you can see the external stress of a cell. The cell can be disrupted. That can be protected by what you call that? Intermediate filaments. Okay? So something like that, they give the strength, right? Sometimes you can see some of us build up their body, isn't it? A lot of muscle and then strength and then 6-pack, 7-pack, 8-pack, like that, right? Okay. For me, only the tummy is there. So when you give one punch, I will die, right? So some fellows are very strong, isn't it? Then they are having that 6-pack, 7-pack, right? They can withstand that external stress and the strain. That is the simple example for the intermediate filaments. A cells can be the strength of the cell by external force or stress, it can be disrupted, protected by the intermediate filaments. Microtubules, another kind of what you call the cytoskeletal elements that is inside the cell, internal transport. For example, when we look at this way, when we want to go to what you call Katunayaka airport, right? How do we go earlier from Colombo to Katunayaka? We had to go through Paliyagoda, Vattala, Handana, then Siduva, Katunayaka, like that we go. So a lot of traffic, there are a lot of traffic in a sense, we will be end up in 2-3 hours, we will go. But now what happened? We made a highway, isn't it? From Paliyagoda itself, the highway, 25 minutes, we can reach the Katunayaka airport. So the microtubules are like highways inside the cell. The highways inside, this is beautiful, isn't it? So highways inside the cell in a cell. Now suppose we are producing some protein in the of endoplasmic reticulum. And we have to send that protein to the Golgi apparatus, because Golgi apparatus is the one modify the protein and then export to the packaging, we call it. Package karla, post karna. You get the postman job. So the postman is the vesicle. So what happened? Initially, what happened? From the ribosome, the protein is collected in a vesicle, and then that is transported along the microtubules. Along the microtubules, the tube-like structure. On the surface, there are some other protein. I will teach you in the cytoskeletal, and then go to the Golgi apparatus. Understand? So that is, again, the movement of what you call the vesicle inside the cell. And the cell itself can move. There is microfilaments called actin filaments necessary for this. For example, you can see, right? We have a nutrition source that you can see. Actin have in the right hand side, you see the picture, right? You can see actin can be in two formats. One is called small soluble subunits and large filamentous polymers, right? So what happened now? Your food source is the yellow color arrow. You can see this is yellow. Here's the food is there. So what happened? This filamentous actin will become small molecule, right? So disassembly, we call disassembly. They disassemble. The filamentous actin will disassemble, and they will go to the other side of the cell, because the food source is here, isn't it? And they will assemble here. When they, you can see my cursor, assemble here, and then for the filamentous. So when they are the filamentous, and then what happened? That will, the filamentous will grow. Filamentous alkene filament will grow and then surround the food particle and engulf the food particle. And that is simple as amoeba does that. Understand? So this is what we call that. For a cell can change the shape and move, right? And then later I will teach you how the macrophages move, right? From the site of somewhere and then to the site of infection or inflammation, and that site of macrophage undone. So that is the same mechanism. So cell can change the shape and move. At the same time, intracellular transport also can take place. At the same time, cell should not be disrupted and damaged by the external force. There are filaments called intermediate filaments which can protect them. Okay, so that is what you call another function of a cell. And then I told you that cells sense and send information. Right? That is what you can see now. For example, in our cell, we have some inactive enzyme. That enzyme has to be activated. So what happened? The cell has surface receptors. So some of the hormones or molecules comes and bind to the surface receptor. And then you can see that what happened because of the signal. Something like that. For a simple example, I told you. After a meal, glucose level goes up. So we had to reduce the glucose. So what happened? Pancreatic beta cells secrete the insulin. That insulin molecule comes and binds to the muscle tissue, asking the muscle to take up more glucose, asking the adipose tissue to take more glucose. So that hormone comes and bound to the receptor, and then the enzymes are activated, and then glucose utilization increases. So the blood glucose drops. So cells sense it. Who sends the glucose? Beta cell. And then sending the information to where? Target cell. What is the target cell? Muscle cell. Understand? And then you can see now some of the hormones can bind to the cell surface receptors. Some can go inside the cell. Suppose you have a girlfriend, and then your girlfriend, you want to meet the girlfriend, but you are not allowed to go inside the house of the girlfriend. So what you do is, get but you can send a message. Nowadays you can send SMS, but what you do is you send a letter through a small boy playing there. Understand? And then the message will be given to your girlfriend. So that is, some hormones or some messenger molecules cannot go inside the cell. So they have a surface receptor. For some boys, you can go inside the house of the girlfriend and talk to the girlfriend. Father is not a problem. Dog is not a problem. Mother is not a problem. So that is what you call that. They have a receptor inside the cell, cytosolic receptor. So some hormones can enter the cell and can they action. So that is what you call that. For example, some hormones need surface receptor, some hormones can need what you call can easily go through the membrane. The reason behind that, because some hormones are water soluble. When some hormones are water soluble, in a sense, they cannot go through the lipid bilayer. Some hormones are not water soluble but lipid soluble. They can go through the lipid bilayer inside the cell and exert the action inside the cell. Okay, right. So as you all told you again, yesterday also Dr. Nisanshara told about this, the central dogma of the molecular biology. The information of the genetic material, all the informations are stored in the genetic material, that is DNA. As I told you, that DNA is converted to DNA is what do you call that? Right? Replication. I told you DNA become RNA, how do you call? Transcription. And then RNA, mRNA become what do you call that? Protein synthesis, that is called translation, and then folding of the protein, and then function of the protein. So what do you call it? At the end of the day, we have what you call that? DNA become RNA transcription, RNA become protein translation, and RNA become DNA is called reverse transcription. Understand? DNA become DNA is replication. Protein become DNA is what? How do you call it? One of you? Protein become DNA, how do you call it?
DNA synthesis?
DNA synthesis. Can it happen in the cell? That's what my point is. DNA to DNA can happen in the cell. DNA to RNA can happen in the cell. RNA to protein can happen in the cell. Protein become DNA, can it happen in the cell? Can? I don't know. Who is that? Can you answer this? Okay. Okay. How did my professor taught me like this? DNA to DNA is replication. DNA to RNA is transcription. RNA to protein is translation. RNA to DNA is reverse transcription. Protein to DNA, he asked everybody and then everybody was silent and he said DNA to protein is imagination. Isn't it? So protein to DNA is imagination. Cell cannot do that. In the outside, if you know the protein amino acid sequence, you may synthesize a DNA, but I don't think inside the cell we can do that. It's just an imagination only. Okay. So DNA to DNA replication, nobody will ask that. Protein to DNA is what is the, don't go and write imagination. Okay, right. Anyway, and then I told you another function of the cell. Cell has to grow and divide, isn't it? You can see this is what you call cell cycle. When the cell divide, interestingly, you can see that. Right? Cell division is called mitosis or meiosis. Right? And then if the cell doesn't divide, that is end up in non-dividing cell. So the cell cycle is having various stages. G1, we will be studying in detail in the cell cycle later. RNA and DNA protein synthesis take place. There's another G0 that is called resting. And then there's another S, we call it as DNA replication. G2, RNA and protein synthesis, and then again meiosis. So this is what you call cell cycle. In this cell cycle regulated, if this cell cycle regulated, and then what happened, the cell death also takes place, and that is what you call apoptosis. Right? At the same time, cells, right, undergo what do you call that? The reproduction we are calling, right? It's a female, male, meiosis, and then you can the diploid become haploid in the gametes, right? One type of female gamete and the two types, and the male gamete is two types, and then what happen? Crossing will take place or fertilization will take place, and the female zygote and the male zygote, right? That is what you call reproduction. This is what you call mitosis, and this is what you call meiosis, right? The mitosis, the what you call the ploidy is equal, right? 2n will become 2n, the daughter cell. Here, meiosis, what happened? The 2n become half. That means diploid will become haploid. Okay, right. And we can see cells will be die from aggravated assault or programmed cell death. I told you again, right? Aggravated assault is called necrosis, right? Small bell form the structure. Sometimes when we have a very young age, at the age of 30, suddenly what happened? Corona infect you, your lung function is affected, and you get a pneumonia, and then you can breathing problem, you die. Necrosis, right? Okay. And then what happened? Like me, right? You live and grow old, and then you can see, and then after some time, what happened? I may not be able to walk. Another 10 years time, when you become doctor, I may be a patient for you. I may be a heart patient for you. I may be a, what do you call that, having a, what do you call that, you may operate me for some reason, right? I will come with a, what do you call that, right? Everything. So that is what you call, naturally I am programmed to die after some age, isn't it? That is what you call apoptosis, right? So you can see now the cell, I will be teaching what is the process going on in necrosis, what are the process going on apoptosis, but at the moment you know this, cells can be die from aggravated assault or internal program. Aggravated assault tamai necrosis. Internal program tamai apoptosis. Okay. Right. Now all this time we are talked about this. Now we are coming to the components of the cell. Components of the cell, when you look at the components of the cell, right, we have cell membrane, and then surrounding cytoplasm and the nuclear compartment. And the cytoplasm is divided into two compartments, two parts, we call it as cellular organelles and cell inclusions, we call it. Cell organelles are what? Membrane bound cell organelles are there. Endoplasmic reticulum, Golgi complex, mitochondria, plastid, lysosome, you name it. Non-membranous organelles are there. Ribosome, cytoskeleton, centrioles. You can ask a question, MCQ, which of the following organelles are membrane bound? Okay, we can give endoplasmic reticulum, Golgi complex, ribosome, cytoskeleton, centrioles as answers. So you had, this is a true or false. Typical true or false question, isn't it? You got the point, all of you? Are you all there, please?
Yes, sir. Yes, sir.
Yes, sir.
How many of you are there? Mama itu mama nidaganno dan nane meke makara. It look like a, what do you call that, Hindi film with English subtitle wage the nane.
No, sir.
Oh, sir. Understand, right?
Yes.
No, sir.
Okay.
Interesting, sir.
If you feel it's a Hindi film with the English subtitle, tell me, okay? Okay.
Okay. Okay, sir.
Okay, sir.
Okay, sir.
Right. Okay. So now we go to the cell inclusions, right? Cell inclusions are what? Storage products are there. Plant cell starch, fat will be there, oil will be there, proteins will be stored, right? Secretory products will be there, enzymes, hormones, pigments, alkaloid, resins, gums. Excretory products will be there in the cell. Inorganic salts, latex, tannins, essential oils, depending on what type of cell is that, whether it is plant or animal cell. So cytoplasm is divided into two compartments or two categories, cell organelles and cell inclusion. Cell organelles divided into two categories, membrane-bound cell organelles and non-membrane cell organelles. Okay. Right. There is a mechanism, subcellular fractionation of a tissue. Then we can do it as an experiment, but we'll see whether we subcellular fractionation of a tissue. We take a tissue and then there is an instrument available in the lab. We call it as homogenizer. Understand? Using that homogenizer, we can homogenate the tissue. Tissue homogenizer, we call it. Okay. So after tissue homogenate, what we do is we can centrifuge it. There is what you call an instrument in the laboratory called centrifuge. And then that centrifugation, we call it as a differential centrifugation. That means we change the speed and the time, 1000 G, the speed of the centrifuge for 10 minutes. You get some pellets in the bottom. In that pellets, you get nuclei, cytoskeleton, and plasma membrane. Interesting, isn't it? So you decant it, and what you get in the bottom is what? Your nuclei, your cytoskeleton, your plasma membrane. And then you take the supernatant, what is on the top is called supernatant, and you centrifuge again, 2000 G, 20 minutes. And then still some pellets come in the bottom. These pellets contain mitochondria, lysosome, and peroxisome. And you can see, you take the supernatant again, and you go for 80,000 G for 1 hour. You can get microsomes, endoplasmic reticulum, and small vesicle. And then finally, you go for 150,000 RPM, right? And then you get in the bottom whatever pellets containing ribosome, large macromolecules, and then the supernatant will be having a soluble proteins. So all in all, when you look at, you can see the cellular contents can be fractionated and isolated. Understand? Fractionated and isolated. So this technique is called differential centrifugation. Right? Then we have one called isopycnic sucrose density centrifugation. In a sense, different density gradient what do you call that solution, and then you can see the different level. You can see that cellular components will be separated, and then we can, by fractionation, we can collect different fractions, and then different fraction will have different cellular components, right? So what I'm trying to say here, right, we can separate the cellular organelles into various parts, and then that is the way they studied the functions of each and every cellular organelles. Understand? So simple as very easily. For example, I will tell you that when I was doing my postgraduate degree, I isolated beta cell. You know, so single beta cell you can isolate. So you can see the beta cells are, you know, your A-level islets of Langerhans. In the islets of Langerhans only you have the beta cells. So what you have to do? First you have to take the islets of Langerhans. There is a collagenase pavichi karala or digest karala. You can pick up one beta cell using the high-powered microscope. Anyway, that is different altogether. So what I want trying to say is the cellular components can be separated. Okay, understood this part? Okay. Wow, this is the component of cell. This is where we are coming to the real cell now. Okay, so I put this slide. You can see everything. One look, you can find out everything. Understand? So you can see the plasma membrane made out of lipid bilayer and membrane proteins. And then you can see that is the first one. And then as you go inside, the second one is what? Cytoplasm, right? Cytoplasm consists of two components. One is called cytosol and organelles. Cytosol is the liquid portion of the cytoplasm. And that is what you call simplified matrix containing the nucleus and other organelles. And number 3 is the mitochondria, is organelle, which energy is extracted from the food during the oxidative metabolism. And then you have a secretory vesicle, sorry, number four, cytoskeleton, right? That is the skeletal system of the cell. I told you microtubule, intermediate filaments, actin filaments. We have different functions, right? And then microtubule is a tube of a protein molecule that involve in the, what do you call that, transport of molecules inside the cell. And then the structure, something like that, centrioles, cilia, flagella, all these are made out from microtubule. Then we have something called intermediate filaments, intertwined protein fibers that provide the support and strength to the cell. Actin filaments, twisted protein fibers that are responsible for the cellular movement. Understand? So you can see that is number 4. Number 5, nucleus. Interesting, isn't it? You can see, I put one slide like this because one look, you can see everything, right? So command center of the cell. Oh, this is the nucleus, right? So that's the command center, because in this lecture, I am the commander, isn't it? Whatever I am saying, you are listening. Okay, that shouldn't be actually, but command center of the cell. Nucleolus. That's what you call site where the ribosomes are produced. Nuclear envelope, that's a different opening. Nuclear envelope is a double membrane between the nucleus and the cytoplasm. It's not a single membrane. And then the nucleus membrane has pores, nuclear pore, embedded with proteins that regulate passage in and out of the nucleus. You can see either side of the what do you call that, nuclear pore, there's a protein. And then you have the rough endoplasmic reticulum. On top of the rough endoplasmic reticulum, you have what you call ribosomes, right? The ribosomes are the place where the protein is synthesized. And then you can see the beautifully, the the smooth endoplasmic reticulum. They don't have the ribosome, right? There is what is happening in the system of internal membrane, that manufacturing of carbohydrates and lipids, and then tacking of proteins taking place in the Golgi apparatus, right? Golgi complex, we call it. And then something called peroxisome we have. Golgi complex collects the packages and then distribute molecules manufactured in the cell. And then we have peroxisome, that is different from mitochondria. Vesicles that contain enzymes that carry out particular reactions such as detoxifying potentially harmful molecules are detoxified. I will be teaching you about the detoxification in the what you call xenobiotic metabolism. And then you can see the lysosome. Lysosome is the suicide back of the cell, breaks down the macromolecules which are not useful anymore, right? Mutated proteins, deformed proteins, and then matured organelles, all are destroyed by lysosome. So these are the components of the cell: membrane, cytoplasm, cytosol, and organelles, and different organelles and their different functions. Okay, right. So we first look at this, what do you call that, cell membrane, right? When you look at the structure of the cell membrane, right, basically when you look at the structure of the cell membrane, right, it's beautiful, isn't it? This is what you call fluid mosaic model. I am not going to talk about how this history developed to finally to end up a model called fluid mosaic model. It is postulated by two scientists called Singh and Nicholson in 1972. That is even after I born. Okay, so the membrane, right, it's enclosing the cell, isn't it? So that is what we call it as the enclosed structure surrounded by this plasma membrane. There are two components we have for this membrane structure. One is called structural component. Other one is a functional component. The structural component is phospholipid, right? When you look at the phospholipid, Dr. Nisansala yesterday talked something about the phospholipid, and she will be doing more in the lipids. It has what you call that a hydrophilic head group, and then there are two tails that is coming from the fatty acids. That is a hydrophobic, hydrophilic. Philic means liking, water liking, and then phobic means water disliking, phobia. So the hydrophobic tail and then hydrophilic head, and either we call it as a non-polar tail and polar head. So what happens? This phospholipid will be arranged in a bilayer, exposing the hydrophilic head to the aqueous environment outside, as well as exposing the hydrophilic head, the aqueous environment inside the cell. Understand? So the middle part of the bilayer will be a hydrophobic nature that will consist of the tails of the phospholipid. The tails of the phospholipids are the fatty acids. Okay, right. So you can see embedded in the cell membrane, there are what you call proteins. There are two kinds of proteins are there. One is we call peripheral proteins, that is located on the surface of the membrane. Understand? And then some proteins are, we call it as integral protein. So the proteins are two types, peripheral proteins and integral protein, and carbohydrates are attached to this protein. When the carbohydrate attached to the proteins, we call it as glycoprotein. When the carbohydrate is attached to lipids, we call it as glycolipid. And then the carbohydrate component present in the cell membrane is generally called as glycocalyx. And in between the double layer, there is another molecule called cholesterol. Yesterday, she mentioned about the steroid ring structure of the cholesterol. Cholesterol also amphipathic molecule. And then we will be talking about the amphipathic molecule. What do you mean by amphipathic molecule means, right? If a molecule having polar nature and then non-polar nature, that is called hypoamphipathic. If a molecule have a hydrophilic group as a hydrophobic group, that molecule is called amphipathic. So phospholipid is an amphipathic molecule. Okay. Likewise, we have what you call that our cholesterol molecule is there. Cholesterol molecule, if I can remember, I think 1, 2, 3. There are 3 or 4, 6 member ring, and then there's 1 5 member ring, and then there's a what do you call that side chain. That's cholesterol molecule. She will be doing in the lipids in detail. It also has a OH group. Cholesterol molecule has OH group that can have a polarity, and based on that polarity, cholesterol molecule also having hydrophilic as well as hydrophobic nature. That will be in between this bilayer, lipid bilayer, and then the purpose of the cholesterol molecule is what you call that, giving fluid nature to the membrane. I will come to that in a while. Why we call it as a fluid mosaic model? Because these components can move from one place to another place. And then you can see this bilayer, what happened, these proteins are embedded. When the proteins are embedded, it's like a mosaic appearance. That is why we call it as fluid mosaic model. That's why we call it as what? Fluid mosaic model. Okay, right. So, cell membrane are fluid mosaics of lipids and proteins. Phospholipids are the most abundant lipids in the cell membrane. Phospholipids are amphipathic molecules. Huh? So the membranes, hydrophobic and hydrophilic regions, right? The fluid mosaic structure model states that membrane is a fluid structure with mosaic of various proteins embedded in it. The phospholipids in the plasma membrane can, right? There's a word is missing, can move within the bilayer. That is called lateral movement of a bilayer. Most of the proteins and some protein can drift laterally, drifting from one place to other place. If I get phospholipid drift and from one monolayer to another monolayer also phospholipid can go, but very rare. That movement is called flip-flop movement. Right, you can lateral movement one left hand side phospholipid can be replaced by the right hand side phospholipid. They never maru end up. That is what you call lateral movement. 10 to the power 7 times per second. So it's a very rare occurrence. Sorry, it can happen, sorry. Right? And flip-flopping across the membrane. This is keep continuously happening. 10 to the power 7 times per second means every time that lateral movement is happening. Understand? From one monolayer to another monolayer also the phospholipid somersault karla end up. So that is what you call flip-flopping. The flip-flopping can happen once per month. So you can see when this lateral movement of the phospholipid and the drifting of the proteins in the embedded lipid bilayer, then what happened? There will be a wavy movement like appearance of the membrane. That is why it is called fluid in nature. Understand? Okay. Then when you look at this, some time back, you can see there was a TV advertisement, right? That is what for the Yellow Pages. Yellow Pages TV advertisement, and then you can see most of the time what happened. You can see 1, 2 fingers they show, and then that they move, right? Tuck, tuck. That is what you call go through the pages like that. Yellow Pages advertisement. So that is like that. The tail wax. Right? It wax. It is not stagnated there as it is. Understand? So when you look at this again, right? The temperature cooled membrane, it can from the solid state to fluid state to solid state end up. Or membrane liquidity of the membrane is determined by the what do you call that fatty acid. And a phospholipid gattut, or a phospholipid backbone is glycerol. You all know glycerol, your A-level knowledge. Glycerol is a trihydroxy alcohol, isn't it? Again, then 3 OH groups are there. 2 OH group is esterified with the fatty acid, and 1 OH group is not only esterified with the fatty acid, but there is a phosphate group also, and then together with another compound is attached. So here, sorry, 2 tails and then 1 phosphate is linked to with another molecule that you will study in detail. So you can see the phosphate is negatively charged. That is why the head is hydrophilic. The tail is either it is saturated fatty acid or unsaturated fatty. The double bond in the hydrocarbon chain, double bond is the double bond is there, that is unsaturated. Single bond is saturated, simple. So saturated fatty acid tibunot, the liquidity is very less. Membranes rich in unsaturated fatty acids are more fluid. At the same time, the steroid cholesterol have different effects on membrane fluidity in different temperature. Right? In the warm temperature, right, cholesterol prevents and then restrain the movement of the phospholipid. At cool temperature, it maintains fluidity by preventing tight packing. So the cholesterol has 2 roles, right, in the maintaining the membrane fluidity. So when you look at this picture, you can understand when you have the unsaturated hydrocarbon, as Dr. Nisansala yesterday said, whenever you have a double bond, there's a bend. That is what you call king formation, right? So when you have a king formation, when the tail bends, the packing is impossible. That is one of the reasons the fluid nature of the bilayer. They are not compactly packed. Understand? When you have what you call the saturated hydrocarbon, they are, you can see, it is like a viscous nature, more solid. That means the tails are, there's no king formation in the tail, so they are packed compactly. So you can see that with the temperature, whether a membrane has to be in fluid nature or solid nature, when the temperature increases, the membrane will become more fluid. That is prevented by cholesterol. On the other hand, when the temperature is less also, the freezing condition, the membrane become more solid. That also prevented by what? Cholesterol. So the cholesterol has a dual role. Either in low temperature or high temperature, regulate the membrane fluidity. That's the role of the cholesterol molecule. Okay, so this is all about membrane. So when you have a protein molecules are arranged in two different ways, peripheral proteins or extrinsic proteins, we call it, right? And then they are present in the outer and inner surface of the lipid bilayer. So peripheral protein usually located on the surface. And the integral protein or intrinsic protein, we call it. Peripheral protein, there's another name called extrinsic protein. The intrinsic proteins penetrate the lipid bilayer partially or wholly. Right? So when the integral protein span across the membrane, you can see the membrane bilayer, there is a outer membrane domain. There's a transmembrane domain. If you look at the picture again, you can see that basically. And you can see now that this membrane, right? Okay, this is the protein, right? This is the integral protein. You can see my cursor. The outer portion is what? Outer membrane domain. The inner membrane domain, what is inside the cell. And then there's one that is called spanning the double layer. That is what you call transmembrane component. So a integral protein spanning across the membrane, that we call it as what you call that transmembrane protein. Okay, right. So the peripheral proteins are bound to the surface of the membrane. Integral protein penetrate the hydrophobic core. Right? Okay, right. So you can see the later you will study, Dr. Delaney will teach you protein structure and amino acids. There are the protein, you can see there is the transmembrane protein. Imagine transmembrane protein are the oluwa. We have a bilayer එක උඩින් තියෙනවා කැලක්, bilayer එක ඇතුලෙන් තියෙනවා කැලක්, across the bilayer also කැලක් තියෙනවා. So the outside the membrane තියෙන එක outer membrane domain, across the membrane transmembrane domain, but is inside inner membrane domain. Also cytoplasmic domain, we call it. One is called extracellular domain. What is outside the protein projecting outside the bilayer called extracellular domain, right? And then protein projecting inside the cell is called intracellular domain. And then the protein across the bilayer we call transmembrane domain. Understand? So you can see amino acids also, there are hydrophilic amino acids, hydrophobic amino acids, like that. So most of the amino acids located on the extracellular domain of a protein, of an integral protein, or amino acids present in the intracellular domain of the transmembrane proteins are mostly hydrophilic. The amino acid making the part of the protein in the transmembrane domain is mostly hydrophobic because that is embedded in the hydrophobic layer of the lipid bilayer. So hydrophobic interaction, hydrophobic hydrophilic cannot be together. Outside the cell is aqueous, hydrophilic. Inside the cell, aqueous, hydrophilic. So you should have hydrophilic domain outside and inside. So you should have hydrophilic amino acids making the protein outside and inside. And then hydrophobic amino acid making the portion of the transmembrane protein. That is what I showed you in this. Okay, so you can see now the beautiful structure of a transmembrane protein, isn't it? Usually you can see that intracellular. Hello.
Hello.
You are there, right?
Yes.
You're not watching any YouTube movie, right? You're listening this, right?
Yes,
Yes,
sir.
sir.
Yes, sir.
අනේ මොකද මේ උදේ පාන්දර කෑවේ නැත්ද මොකද්ද? හා? 18 100 වික්ක වගේ ඉන්න ඔක්කොමල මොකද්ද? හිනාවල එන්නකො නිකම් පැතොලොජි මුණ පෙන්වන්න එපා. හරිද? මට බයෝකෙමිස්ට්‍රි මුණේ හිනාවෙන් ඉන්න ඕනේ. හරිද? Okay. Right. Active වෙන්න ඕනේ. හරිද?
Okay, sir.
Okay, sir. තමයි. Hurry. So you can see now the bilayer is there, and then you have the protein, right? You can see the proteins are nicely coiled, and this is alpha heli. Dr. Delaney will teach you. So you can see extracellular domain, and you can see the intracellular domain, and you can see the transmembrane domain beautifully. The transmembrane domain of the proteins are made out of hydrophobic amino acid, and the intracellular domain of the protein is also made out by the hydrophilic amino acid, and the extracellular domain is made out by the hydrophilic amino acid again. So you can see that N terminus is outside and the C terminus is inside of a transmembrane protein always, right? So this is a beautiful example for a transmembrane protein that is the functional component of the cell membrane, peripheral protein and the transmembrane protein. And then what are the functions we are going to do? So membrane protein, somebody asked you a question, what are the functions of a membrane protein? Then what we will say, transport, enzymatic activity, signal transduction, cell-cell recognition, intercellular joining, wow, attachment to the cytoskeleton and extracellular matrix. Interesting, isn't it? So this can be an SCQ question. List 6 major functions of membrane protein: transport, enzymatic activity, signal transduction, cell-cell recognition, intercellular joining, attachment to the cytoskeleton. Okay, right. So the plasma membrane encloses the cell contents. It provides cell shape, characteristic shape of the red blood cells, nerve cells, bond cell, etc. And then they are selectively permeable. I'll come to that in a while. So you can see some of the functions of the transmembrane proteins are given here. So you can see the first one is involving the transport. You can see that protein spanning across the membrane, it acts as a carrier protein, transporting, right? Like forming a channel, right? So the main transmembrane protein can form a channel. Through this channel, what can happen? Ions can be transported, right? So this is called ion channels, right? So that you can say ions are protected from the hydrophobic region of the bilayer, right? These ions can be transported in and out, understand? And then they can act as, at the same time, they act as a carrier protein also, right? Carrier protein also means, that means they don't form a channel, but what happens? The molecule comes and binds to the surface outside, and then this transmembrane rotates, and then outside portion will become inside part will become outside part, the molecule will be transported inside. Understand? So that is what you call carrier mediated transport. The protein act as a carrier. The protein can act as a channel in the transport. That you can see, some of the membrane bound proteins act as enzyme. You would have done your A-level electron transport chain. The electron transport chain, most of the enzymes are bound to the cell surface of the inner mitochondrial membrane. So you can see there is the blue color one is the substrate, and then the red color one is the product. Here what happens? Enzyme act as what you call that enzyme, the protein. And then cell surface receptor, that is what we call that. For example, it cannot, the molecule, the signal molecule cannot go, the hormone cannot go inside the cell. So it comes and binds to the surface. And then there is some intracellular mechanisms are taking place. That is what you call signal transduction. That is what you call second messenger. Now, I told you, right? You can't go inside the house to meet your girlfriend. And then you are the primary messenger. So you find a small boy playing in and around the gate, and you pass the letter to your girlfriend, and that boy goes as the second messenger. So what happens when the hormone comes and binds to the top? Some molecules are formed inside the cell. These molecules are called second messenger that will give the message what cellular action has to be happen. So that is what you call signal transduction mechanism or second messenger system. The hormone is the primary messenger, and then the red color molecule is the second messenger. And then you can see the lipid bilayer, right? The transmembrane protein is located as a cell surface receptor. It's simple as that. Now, for example, why COVID-19 is attacking us, not any animals. Unfortunately, our human karma or whatever we did something bad to this earth, and then God is punishing us. Unfortunately, there is spike protein in the virus. The spike protein in the virus can fit into the receptor located in our lung cells. Understand? So the virus is transmitted inside. None of the other animals having that. Understand? So the protein can act as a cell surface receptor. And then you can say cell surface identity marker. Cell recognition, we call it. For example, cells together form the tissue. So what happened? One cell can recognize another cell and then identify it as a same type of cell and form the tissue. Sometimes what happens? Cells can reject another cell. That is what you call rejection. So that is what you call cell recognition. This carbohydrate portion attached to the protein act as what you call that cell recognition molecule. Cell addition, you can see now. One cell can join with another cell through protein. That is what you call cell addition. You can see inside what you call that, we have a skeletal system that is called cytoskeleton. You can see what the cell membrane can attach to the cytoskeleton via some other proteins. This is simple example for RBC. I will teach you in the cytoskeleton. So attachment to the cytoskeleton, again, another function of the transmembrane protein. Okay, so you can see that one by one, I have given the notes for that. Transport, enzymatic activity, signal transduction, intracellular joining, cell recognition, and attachment to the cytoskeleton and extracellular matrix. These are the functions of what you call that transmembrane protein. Okay, so now the time is 10 o'clock. I will stop here and then we have another tutorial. Understand? I think this week somewhere we have a tutorial on function. This one, right? Hello?
Yes, sir.
Yes, sir.
Metma, are you there?
Yes, sir.
Yes. Hayyen kata karanako මොකද කෑවේ නැත්තේ?
Yes, sir,
Hello?
I am there.
Yes. When are we having the next tutorial on this section? No idea. You didn't have the timetable.
On Friday.
On Friday, right?
On Friday, sir.
Yes.
Yeah. That is also 2
10 days.
hours, isn't it? That's all 2 hours, isn't it?
Yes, sir.
Okay. 1 hour I will do the rest of the slides and then 1 hour I will do the tutorial. Okay. Everything fine?
Okay, sir.
Anything you want to ask? Am I too fast?
No, sir.
Am
I
I
have
too
a
fast?
question.
Am I too fast?
No, sir.
No, sir.
Am I too slow?
No, sir.
No, sir.
Is it look like, is it like watching a Chinese film? No.
No, sir.
No,
Is it like watching a Hindi film with subtitle? No.
no, sir.
Okay, that's how it is. Anyway, so any question you have?
Sir, can you hear me?
I want you to talk loudly. I can't hear you.
Sir, can you please explain peripheral proteins and integral proteins?
Peripheral protein and integral protein, right? Peripheral protein, you can see when you have a lipid bilayer, there is a two monolayers are together and form the bilayer. Harida? Okay, right?
Okay,
And then peripheral protein mean the protein attached only to the surface, either outside surface or inside surface. Okay, it cannot penetrate the membrane bilayer penetrate කරන්නේ නැහැ. තේරෙනවද? So attached, temporarily attached, it can be removed easily. But when you come to the transmembrane protein, integral protein, it span across the membrane most of the time. So it penetrates the bilayer. So when it penetrates the bilayer, outside also it has a domain. Inside also it has a domain. That is called outside is extracellular domain. Inside is intracellular domain. And in between the bilayer also there's a domain that you can see that in the picture I showed you. You can see now this is an example for peripheral protein, like this span across the membrane, right? So there's an outer portion, there's an inner portion, and there's a transmembrane portion. But when you look at the peripheral protein, it will be on the surface, outer surface or inner surface. That is, you can see that in the other picture I show you. Now you can see now, this is actually a peripheral protein that it doesn't go inside, only located. Now this is a peripheral protein. You can see inside the surface. And then what is yellow color is a cytoskeleton. And then this is the transmembrane protein. So the peripheral protein is only in the surface, not inside. Not across the bilayer. Any other question?
So what do you mean by the domain, sir?
Domain means then that you will study in the protein. Then we can go. Then always your head is one domain. Your leg is one domain, part of the protein. Understand? Okay. Domain means part of the protein. There's a definition for the domain. Dilini will teach you. Domain is one part of the protein, right? Okay. At the moment you understand is a part of the protein is the domain. Intracellular domain means the part you find inside the cell. Extracellular domain means the part find outside the. Okay.
So what are the references?
Yes.
So what are the references book?
What are the references book? You can read any biochemistry book for this. It's given to you, right? Okay. And then I think most of the time my notes is adequate enough. And you can read a book also. You can read any biochemistry book. I mean, you don't need to go into detail of these actually, as far as medical students is concerned. Whatever I told is adequate enough. You don't go into depth of it and then try to. There is a disease called that, and first year students keenness very, you are very keen, right? So you want to read everything. So that is, I always tell the students, you are having a disease called hyperkinemia. Understand? So don't get a hyperkinemia letter. Whatever the objectives and then right? You can read any biochemistry book, whatever Lippincott also you can read, and then you can read Harper, you can read any book, you can read. All the books will be giving the same ideas. Anything else?
Sir,
Yes.
sir, lysosomes and peroxisomes are specialized vesicles, right?
Lysosome and peroxisomes are specialized. Peroxisome is involving in synthetic reaction, but lysosome is not involving in any synthetic reaction. Lysosome is a suicide bag, and then inside the lysosome you have some enzymes are there. These enzymes are the one. If the lysosome leaks, what happens? The cell will die because this enzyme. The pH of the lysosome is 4.5. I will teach you when I come to lysosome. I will teach you. Right, the pH of the lysosome is 4.5. The cellular pH is 7. Right, so the enzyme kept inside the lysosome is 4.5, and then usually what happened, the older older organelles, and then the defective proteins, and then the matured proteins are destroyed by the enzyme present in the lysosome. Lysosome doesn't involve in any synthetic reaction, but peroxisome is involved in some of the synthetic reaction. I will teach you when I teach you peroxisome.
Okay, sir. So are other vesicles considered as organelles, like secretory or transport vesicles? Are they organelles or just?
You can see now, I had a classification, right? Come on, Pute. You have to be very attention, no? Me balandako. Be something we call cell organelles, something we call non-membrane, and something we call cell inclusions. The secretory products, right? Enzyme vesicles, hormones comes in vesicles. These are we call cell inclusions, isn't it? Right? These are not considered as what is, if a protein is synthesized and packaged in a, what do you call that, vesicle, that is not an organelle actually. That is the vesicle is formed to transport this thing. That is Golgi apparatus is a organelle. Understand?
Yes, sir.
Okay, so those are called secretory vesicles. Secretory vesicles are not considered as organelles. Okay,
Okay, sir.
they are
Thank you.
having just having the package and transport function only. Anything else, ladies, girls? Silent. How do you feel now? How do you feel? Aggressive attack the aggressive attack means sudden cell death the necrosis the. How do you feel?
You feel good, sir.
You feel good or slow death. So you will end up in apoptosis, not necrosis, right? Okay, you have to give the feedback.
So, if that's a you told, sir.
Yeah.
Sir, question.
Katha karanda.
So you told that that transmembrane protein it changes the side to transport.
Transmembrane
What is
protein.
that, sir?
Transmembrane protein is usually involved in transport. Understand? Okay. One of the function is transport, not all the functions. There are 6 functions I have given, one transport, right? So some of the substances from cell inside has to go outside, waste products. So some enzyme has to go outside, some hormone has to go outside. So what happened? They cannot go through the bilayer. Bilayer is not diffusible through all the substances. Small molecules, lipid soluble substances can cross the bilayer. So you need some molecules which can transport across the membrane. So that is what these proteins does. The transmembrane protein does. The transmembrane proteins together can form a channel, right? Then they are called channel protein, or they can act as a carrier. Then they call it a carrier protein. But then channel protein ions can go through the channel, and then the ions can be prevented from exposed to the bilayer, because ions are hydrophilic now, and bilayer is hydrophobic now. So that the enclosed structure is ion channel, through that ions can go. And some molecules come and bind to the, you can see when a transmembrane protein has what? Extracellular domain, transmembrane domain, and intracellular domain, right? So one molecule will come and bind, and then what happens? This carrier protein will rotate. So the outside part will go inside part will go outside. So what happened? The outside again, bind will have to be a substance at retain. That is what you call transport. Okay.
Why is difference between the channels and carriers? Different.
What's the difference between the carrier? Channel hara ions, channels hara ions and other molecules go through the carrier. Because ions are ions, no, they cannot go through the bilayer. So what happened? The ions has to be protected from the bilayer, so it goes through the channel, ion channels. Understand? But carrier, any other molecules can bind to the carrier protein and be transported. Not necessary. What do you call that? Channel form current avashiyan.
So these are the channels and carriers are the protein complexes or proteins.
Protein complexes. Several proteins can, subunits of protein can form the channel. For 2 or 3 protein molecule can combine together and then form a channel. I will teach you when the ion channel comes. Understand? Again, the ions goes through that. Okay.
Okay, so understood. Yes, sir.
Any other question? No. So you have another lecture at 10:30. So shall I stop here then? So we'll meet on Friday.
Okay, sir.
Yes.
Okay, sir.
Okay. Everybody okay?
Okay, sir.
Right. Okay.
Okay,
Bye. Take
sir.
care.
Thank you.
Thank you.
Thank you, sir.
Thank you.
Bye.
Thank you, sir.
Thank you.
Thank you, sir.
So this recording has
Thank
to
you,
be-
sir.
Yes. Wait. 1 minute, and I have to stop the recording, isn't it?